Okay, great. Thank you. Yeah, I work for the strawberry plant breeding program in Puyallup for WSU. And I want to give today some preliminary results from a study looking at different plug types for fresh strawberry in Washington and the Pacific Northwest. So as Lisa mentioned, this study is part of a larger project looking at various aspects of different techniques, methods, and hopefully resources that can become available to fresh market growers to support them in their ability to produce high quality strawberries for those local markets that we were talking about with the panel, just hitting those demands and then hopefully maximizing the ability for us to meet those demands for fresh strawberries. So what is the problem we are hoping to address? The main thing is that to grow for fresh market, multiple people, Lisa DeVetter, Tom Walters, Eric Gerbrandt in British Columbia have shown that planting fresh strawberry cultivars in the fall, we're talking late August or early September, really helps the plants to establish and then to produce those early and high yields starting in the following year. The issue with that is that by the time that growers want to plant in the fall, the plants that are available are frigo plants that have been harvested the previous year and then held in cold storage for 8 or 9 months by that point. So what would be ideal is if there were plug plants that were economically priced that farmers could utilize for hitting that fall planting window. So producing plug plants in a conventional way would require using mother plants and then potting them up and then pegging runners and producing them into a greenhouse plug. Uses a lot of greenhouse space, is expensive, and often to just make them economical enough, each mother plant would have to produce, I heard one estimate, 70 runners for it to become a process that growers could buy into at a not prohibitive price. So the question was, can tissue culture, some aspect of tissue culture, provide an opportunity for those plug plants to be produced without having to take up all that greenhouse space? And another word for tissue culture is micropropagated plants. Those are plants that are produced in a sterile, artificial medium that you can then propagate them up in small areas and then move them into the greenhouse to become plug plants for the field. And that could cut time and also help control viruses and have a very clean stock that could then be transferred into the field. One issue with that, there's no question that tissue culture can produce large quantities of very healthy, vigorous plants. But the issue is that sometimes when those plants are moved into the field, they behave strangely. Some studies have shown that those plug plants from tissue culture flower excessively, producing a very small fruit or misshapen fruit. A few have shown an excessive tendency to runner, which creates a problem for the grower then as well. Those studies that have shown negative effects of tissue culture on field performance that seem to be dependent on which cultivar was being used and also how long it was in tissue culture in that artificial medium. And there have been several studies that showed that plants coming from tissue culture or that have been runnered from tissue culture had no statistical difference in yield or other field performance factors. So our objective was to look at Albion, which is the most widely planted fresh market cultivar in the Pacific Northwest. No studies have been published on the field performance of tissue cultured Albion. So we want to look at Albion and to see if using tissue culture in some process of the plug production systems in the nursery could be a viable alternative. So we produced 3 different kinds of plugs. One was a straight up tissue culture plant. So we produced it in the artificial sterile medium and then rooted it in the greenhouse to produce what looked like normal greenhouse plugs. And so we called that just a straight up TC plant. The second type was a runnered plant from a tissue cultured plant. So where the mother was a tissue cultured plant, and then we took the runner off of those to produce plugs. And then the third type was a runner from a bare root plant. So we planted frigo plants in the greenhouse, took the runners off of those, and developed plugs out of those. So we planted those in replicated plantings in Puyallup, and then evaluated yield, fruit size, and runnering. So I'm just going to go through the preliminary results. Again, this is 1 year of data. So we're hoping to look at these plugs, how they perform for a second harvest year. Looking at yield for that 1 year, we did not see statistical differences in the yield. So statistically, they all yielded similarly. You can see there's quite a bit of variability.